Welcome to the webinar series by IITACB. So IITACB, as we know all, that unique platform to enable networking amongst IIT alumni and also to establish collaboration between IITs and industries. So IITACB is established by IIT alumni from across all IITs and recognized by the MHRD. So for today's webinar, we have our speaker, Professor K.S. Narayan. So Professor Narayan obtained his MSc degree from IIT Bombay, and then his PhD, he obtained his PhD from Ohio State University, USA. And currently he is faculty in Jawaharlal Nehru Centre for Advanced Scientific Research in Bangalore. So he has been active in the general field of molecular organic polymer bioelectronics at GNCSR. And his research led to early reports on plastic optical field effect transistors, polymer solar cells, and polymer light emitting diodes. He has also contributed to the research areas of the soft electronic polymers in the biomedical arena for tissue engineering and vision prosthetic elements. Professor Narayan also works in developing noise measurements and scanning techniques to predict the full life cycle of photovoltaic modules. And he has been the Dean R&D at GNCSR, coordinator of Technical Research Center, funded by DST, an initiative for commercializing inventions. He's also co-founder of a startup, H-Bar Omega. He's a fellow of Indian academies like Indian National Science Academy, fellow of National Academy of Sciences, and he's also fellow of Indian Academy of Sciences. So this talk will be moderated by Dr. T.R. Raju, who is the Director of Research, Sankara Academy of Vision, Bangalore, and he's highly accomplished medical researcher with extensive experience in academic leadership and well-funded research project. He's also a former AS Paintal Distinguished Research Chair, Indian Council of Medical Research, former senior professor and head of neurophysiology, NIMHANS, Bangalore. And he is also former senior fellow, Neurobiology Research Center, University of Sydney, and Dean in an Academy of Neurosciences. And he has proven his ability in managing large public hospitals, including crisis management and grievances redressals, and to serve as an expert advisor to the government and non-government organizations. So this talk will be co-hosted by Dr. Sushila Venkatraman and myself. And for audience, the announcement is please post your questions in Q&A box, which will be taken during the discussion. So over to Professor Narayan for the talk.
Yeah. So thank you, Abba. Good evening, everyone. So I'm going to be introducing or discussing this particular topic, which is really multidisciplinary in nature, truly multidisciplinary. As Abba introduced me, you would realize that I'm not a clinician or a medical doctor. So please don't take my suggestions or some of the statements for any medical treatment and so on. Okay, so that is the first disclaimer. My contacts are written below. So I encourage people who have queries. I mean, some questions, if I've not answered or if you have more questions, please contact me. So for the talk today, please pose your questions in the Q&A box, and hopefully we'll get through it. So the first part of the talk is going to be a long introduction, and then we'll take a break, and then I will show some preliminary results from my lab on some of the topics I'll be discussing. So for me, vision. The process has really been fascinating. It's been my hobby to just make sense out of it or understand it. How a tiny aperture really gets this fantastic set of information, and what we actually interpret as seeing is what we perceive through a combination of the hardware in the eyes and the brain processing unit. Okay, so it's a combination which is at work. Again, for today's talk, I'll be discussing the topic of interest is in the hardware, in the retina aspects, in the right in the front where the photon interacts with the eye. Okay, so the numbers are truly astounding. So I'm an experimental physicist. So looking at photo detectors in market, camera elements, these are numbers which are truly, I mean, perfect in as far as we are concerned. It's as far as the brain is concerned. Okay, so for instance, we perceive, I mean, intensities over very large orders of magnitude. Okay, quite dark to very bright. Okay, spanning nearly 0.1 billion. All this vision, process, and perception, seeing everything takes only very little energy. Okay, 4 milliwatts, but it's always operating. Even the fact that you're alive in this dark, one consumes power at the rate of 2.6 milliwatts. Okay, and the power supply voltage, which is an internal potential which develops when certain things happen, is in the range of millivolts. Okay, so these are very tiny things. Okay? And I mean, and for me, one of the outstanding feature is this capability to detect at single photon level. Okay, the eye is able to really respond to a single photon, and so on. So, okay, the key part or the key point is the photoreceptors, which I'll be just discussing. Please note the numbers. It's in the range of 100 million. Okay, and so you have a circuit which actually is sort of a layered circuit where these the cones out here. And let me turn on my laser pointer. So you have the rods and cones. So what we have is a layered structures. Okay, and the layered structure means you have, I mean, so like this, like in semiconductors, you know, one stacked above the other. So in this case, you have the receptors, then the bipolar, and then the ganglion. The ganglion cells are the neurons, which finally take the signal to the brain using the optic nerve bundle. Okay. In between, you have these synaptic zones of horizontal cells and amacrine cells. Okay. And the architecture is truly amazing because this is sort of preserved across the vertebrates, you know, many animal species, mammals. I mean, this kind of architecture is sort of preserved. There are details in terms of rods and cones which are different, but the signaling mechanism is truly something universally preserved. So this is an important thing from a evolutionary standpoint of view. Now, so some of the abbreviations which may be required to follow today's talk is going to be the photoreceptors. So each, again, I'm showing you the stack of retina. So you have the rods and cones, which I'm going to collectively call as photoreceptors. And then you have the bipolar cells, which sort of has a junction with it takes the input from the rods and cones. Okay. And then you have the neurons, which are the retinal ganglion cells. Okay. So, so RGC stands for truly the neuron, which communicates with the brain through this optic nerve. Okay. And you have some other important cells like these retinal pigment epithelium and macular cells and so on. Okay, everything is important. Everything has a function. And some of the numbers, okay, the numbers matter. So in terms of the different type of cells, there are around close to 60 to 80, okay. What makes this number big, okay, is the subtypes. There are like 20 to 30 different types of retinal ganglion cells. Now, when I say type, it means they are either differing in structure or some very tiny changes, which actually change the function of what this particular neuron is good at. Okay. So classifying these cells eventually is going to be important if you want to give information to them or pretend as if you are giving information from that particular cell to the brain. On the whole, we have around 100 million rods and cones and around a million or so RGCs. Okay. So it is not a 1:1. Okay, it's not a simple correspondence. Only in the center part, the fovea, there is a direct connection. The rest of it is through this horizontal communication which comes in. So it's a very robust architecture which has been, people have tried to mimic it in the lab, in semiconductor labs, and not really been successful. Okay, part of it is because it is silicon. Silicon is dark. I mean, there are many issues with it. So I will come and discuss about those lab-based retina in the later part of the talk. Okay, so what I will not be discussing today is what happens in the brain. Okay, so once the signal goes to the brain, there is the geniculate nucleus, from which it branches out to the actual processing unit in the cortex. So these are things which will not be, relevant for today's talk. So we are going to be focusing on this particular architecture of retina. Okay. Okay. So but I should just, as a small point of digression. So apart from the vertebrates, we have some incredible creatures, especially marine creatures, marine crustacean, this particular example, which is supposed to have the world's best eye. Okay. Completely different architecture. They have 16 photoreceptors, for instance. They can see across the spectrum, UV to near IR. And most intriguing thing is they can detect polarization. Okay, not just linearly polarized, but also circularly polarized light. It's hard to fathom what it really perceives. Okay, and each of them can move independently. Okay, so this is a superb thing to imitate if one wants to design cameras. So we have one recent big gadget from Apple which has come out, which I think pales in comparison with this invertebrate creature out here. Okay, so just for the students to refresh their undergraduate biochemistry, the photon interaction with the retina happens at, the whole initiation happens at the molecular level, where there is a pigment, a chromophore, which absorbs the light. Okay? And then you have a cascade of enzymatic reaction. Okay, and this cascade, okay, it's got a lot of things built into it. Okay, so basically, what happens is you have the chromophore, which is anchored onto a helical structure, which is folded, 7-folded, and these are channels. Okay, so when the molecule absorbs the light, something happens to the channel traffic. Okay, like the Bangalore traffic. Okay, so when you open up something or close with the light, there is a release of ions. In this case, sodium ion channels open and shut when we see the photon interact with the light. So the initial reaction, okay, for the chromophore, which is actually a retinal molecule, there is a structural adjustment which happens at femtosecond time scale. This is truly fast, okay, and one really needs a lot of techniques to actually see this. This has been seen in the lab, and it has been well documented. The entire process has been well documented. So you can see that the photon interaction begins with a time scale at femtosecond, okay, and goes on till seconds. Okay? So there is a whole cycle involved. Okay, so the first step of cis to trans, okay, involves the light, and then the trans actually needs to go back. Okay, that is where the retinal pigment epithelium comes into play to replenish. So this is not a cycle which can complete on its own without an energy source in the background. So that is provided by the adjacent layer of the photoreceptor, which is the RPE layer. Okay. So now, so any problems with these chromophore is quite a challenging thing to address. So there are retinal disorders and diseases which are specific to non-functioning of this particular cycle, which is initiated with light. Okay. So one of the well-known one is the inherited, which is based on the genetic disorder, largely is the retinitis pigmentosa, or under a general class of inherited retinal disorder. Okay. There are various forms of this particular disorder. There are close to 200 genes involved. Okay. And so it is hard to target, so you just can't repair one particular sequence and expect it to function again, because there is a combination involved in terms of the disorder manifesting. And the other one is related to the slow degeneracy over during an aging mechanism, where the receptors actually gradually die because they don't get the fuel, or they are sort of the RPE replenishment doesn't happen at the same rates. Okay. So both these kind of disorders need solution. So just to emphasize, okay, 2 points, I will come back to this. In case of retinitis pigmentosa, unfortunately, the child after is born the images starts getting dimmed out, okay, at very early stages. So it's a very sad, tragic disorder. And the other one is from the periphery center onwards, you start seeing a blocking aspect, which gradually disappears with age, okay? And so you reach a level of blindness from the center to the periphery, okay? So in terms of number, how many actually are affected? So the global average is something like the reported ones for retinitis pigmentosa, like 1 in 3,000 or 4,000. In India, it is quite much larger, okay? The reason being, there are many reasons. One, southern India, especially rural part, is staggering numbers, okay? And it's related to, people have seen it in what they call consanguinity is one of the factors. The males are more affected and so on, okay? So anyhow, I'm not an expert in these particular, either the statistics part or the clinical part, but this is the disease which we are targeting, okay? And so once you start examining these disorders in the lab, one can see that there are structural changes which occur as you start seeing changes in the function of the retina, okay? So the functional changes are, one is the shortening of the receptors. Everything gets contracted, okay? And once that happens, there's an increase in loss of function, okay? And so you are, so it starts at this particular junction out here. Then you start seeing that in case of retinitis pigmentosa, you have an early loss of the rod receptors. Okay, so you get night blind faster, I think. And there are interventions. Okay, this is what I'm going to be just telling you in a very brief way. And the intervention at the early stage is related to gene therapy and genome editing. And slightly at a later stage, even at the early stage, cell therapy has really caught up. This is the stem cell therapy, okay? And there is also a new technique of optogenetic, which is involved, okay, in the early stages. In the final stages, the closing stages, I think the retinal prosthesis really is one of the most promising strategies in terms of vision restoration, okay? While the other ones can prevent, this one, may be able to restore. Okay. Okay. So restoration also is possible with the stem cell therapy and the optogenetic. Okay. So for this is a very quick 20-second introduction to stem. People are not familiar with stem cells. Stem cells are sort of the really potent cells which can morph or differentiate into the functional cells which you want. Okay, and with the right kind of environment and with the right kind of growth factors and chemicals around, you can control in a way you can differentiate into different segments. Okay? And this happens because people understand development quite well and how things get expressed and so on. So one can get these stem cells from either from the embryo, or you can get an induced pluripotent stem cell artificially from some other part. Okay. Both of them can be utilized in this particular case. It has been demonstrated that one can now repeatedly get these RPE cells quite well. There are groups in India who are got this in their labs. And once these RPE cells are, you're able to generate, really it's sort of the breathing apparatus for their photoreceptors. Okay? So you can prevent the early degradation by constantly working on the RPE. Now you can actually grow an entire organoid from these stem cells. Okay, that is the big hot research area where people do 3D tissue culture and can grow an explant. Okay, in the lab. Okay. Now you can take cells from these lab and actually transplant it. Okay, so these are the exogenous sources. So when you do a transplantation, in principle it should work, but and it's been seen to work in some extent. But again, you need to really know how it really fits in, and whether your immune system reacts, whether the architecture is preserved, and so on. Okay. Now there are things which you can do endogenously within the cells. Okay, where some of these macular glia, which I mentioned earlier, they can be made to differentiate into the retinal progenitor cells. Progenitor is the precursor or the mother cells which can mature into one of these functional cells. Okay, so many of these tricks and are known to the experts in the community, and it's really an ongoing hot research area. I have taken for today's talk, this part of the talk, two important references. One of them is this Nature Medicine paper from an Oxford John Hopkins group, which came out last year and a half ago, and another one which I'll be just showing it to you. Okay? So, I mean, the topic is really hot, and I mean, it's difficult to cover. Okay, these are semester courses, but the key takeaway I got from these papers was you can grow the RPE and slow down the photoreceptor cell death. Okay, and so this, in principle, many of these things can happen, and you can transplant. But once we transplanted, at least the photoreceptors, they don't really reach the full morphological maturity. That means in terms of getting the connection and so on, it doesn't. It's a little bit a blind, a roadmap out there. Okay, it's not very clear how you can position and get it working perfectly. Now, so the RGC, the neurons, growing the neurons is the hardest part for the stem cells approach. And this takes, and it is not stable enough. It dies down quickly, and getting the connection with the optic nerve has been shown, but it has been quite challenging. Okay, so this is so we spoke about this stem cell part. So the other important thing is the gene therapy and the optogenetics, which I'll just quickly mention. So there has been a gene therapy based drug. So since we have identified the mutation from the people who have this disorder, this gene has been, you can get it corrected, and you can deliver it using an AAV vector. Okay, so this is a technique which is there. to introduce it into the region of interest where it enters a particular environment and you can get it expressed to get the correct sort of cells with the right genes. Okay. So, and it has been effective in one particular case of liver congenital hemorrhages. Okay. But this has been a much harder thing. I mean, people have tried it over a much longer period, and the success rate has been quite small. But but what has happened is, rather than using it to actually repair it, people have also seen the pathway to the death of a particular cell and try to intervene it using gene therapy. Okay, so there are many levels again and approaches of these gene therapy, both viral based and gene editing tools which have been used. So again, this is a hot area where I'm not the expert, and there is a lot of things happening out here. And I'll just show you the reference where there's a Nature Biomedical Engineering paper which really speaks about it. And the other important technique is the optogenetics, where you can introduce an artificial rhodopsin, a microbial rhodopsin, into any cells, either be it bipolar or the neuron. They are not the same as photoreceptors, but they are sort of artificial from externally derived. And you can make any cells quite photoactive. Okay. Now, unfortunately, one of the problems with this approach is you need a lot of light. Okay. And again, one needs to know this architecture quite well. So this is the important reference which I just mentioned, which came out last month, which tells you the latest in gene therapy and the optogenetic approach. Okay. So, so what we are left with is the prosthetic part. Okay. So in the prosthetic part, what you can forget a little bit of the biology-based technique and view the problem from an engineering point of view, where you're going to assume that if you have a pulsed electrical current or a voltage, which are patterned or is in the form of an electrode, patterned electrode, and you go to the region of interest and give it this particular biases, electrical biases, you can initiate activities. Okay. So the chemical based transmitters, all the biochemical activities can be in some way substituted. I mean, this is also will induce biochemical reactions, which will ultimately do the same job as a natural process. Okay, and now this particular patterned electrode can be stuck at various points, either in the subretinal point where the receptor is, or at the epiretinal point. People have also stuck it near the brain, okay, where the processing is done. So each of these methods has its own complications and difficulty. So this particular program started nearly 20, 30 years ago, okay, nearly in 90s. Argus 2 was one of the first big US-based consortium-led project. You know, hundreds of millions. Maybe I don't know the exact amount, but it's a huge project. Okay, finally, you know, and it involves quite a bit of elaborate camera traits, video processing, and RF signal to the electrodes, which is inserted surgically. And what you get at the end of the day is a vision which is, instead of the perfect vision is 20/20. And in this case, you get a vision of 20/2100. Okay? This is not good. Okay. And this has been FDA approved. Such a treatment, you can see whether a door is opening or shut. Maybe a food is there on the plate, a reflective plate. The idea is to also make people have their own locomotion, or they should be able to walk around, okay, without help. That is rather than see a high resolution image in front. So even out there, I'm not sure whether this actually works. The treatment is in the range of a couple of crores, okay. I mean, the economics are really staggering. But insurance, there are a lot of other issues, okay. And of lately, there are two other projects which have been coming. And the Prima one is the most recent one, which has got the sort of the best numbers. And this is a technology which is from Stanford, now being used by this French company. This is actually quite interesting. Okay, so what you have is a glass in front of you, and then you have a camera which actually gives it, communicates to the glass using an IR source. And then what you have is a set of photodiodes which is inserted on the retina. So the camera converts it into a signal from where you can introduce IR light emitters, which can be incident on the retina. Okay, so it is sort of less invasive. Okay, and the whole thing is you don't need too many cables sticking out, and this looks a little bit more sleek, but again is far from real robust clinical trials. And so, I mean, things are happening. Okay, but hopefully there is a solution. Okay. Hopefully everything works. Okay, that is one of the objectives. So the last thing which I want to do before I break is to show two other approaches. Okay, so this is a hot race from an academic point of view. Okay, the clinical things are far away, but one can see in principle many concepts can really work. Okay, so in this particular case, what the French group here has done is there is an ultrasound or a mechanical sensitive ion channels which has been targeted. Okay, to activate the retinal neurons. Okay, so you use ultrasound techniques near the visual cortex, near the brain, to see if there is light. And there people have seen in these clinical trials or these lab trials, people have seen, perceived the light coming and going as you program it with these ultrasound stimulators. Okay, so there has been a big advancement on the kind of electrical probes which are available now, three-dimensional probes, soft probes, and so on. I will be discussing more about it. And then this may be a good point to just stop for questions. This is an approach which is more like an AI machine learned approach, where if you know all the RGCs, the retinal ganglion neurons on their functioning, one can actually program the electrodes and can convey the information in a more tight way and have it sent to the brain. So this will involve a lot of studies, getting understanding the circuit in its entirety to see whether that can be replaced with processing outside, and whether you can really give these functions right at the RGC levels. Okay, the signals at the RGC. So I hope I've been able to distill the current events which are happening in this field and been able to convey. Otherwise, I can elaborate on anything in these next few minutes. Yeah. Over to you, Raju.
Thank you, Dr. Narayan. For a non-biologist, you've done an excellent job in describing the structure and biology of retina. And I have one question in the Q&A. Can artificial retina be applied for central serous retinopathy? This is the accumulation of fluid in the retina, the vision. Okay. And this is a question.
Okay, can you repeat? What is central serous retinopathy?
Central serous retinopathy is the accumulation of fluid behind the retina. Okay. Currently, you have a treatment available with argon laser. You can do photocoagulation with that. And sometimes it fails. Probably this gentleman wants to know whether artificial retina can be applied for this condition.
Okay. I don't know. Okay. Yeah. I can find that more, and maybe I'll send him an email if he can.
Yeah, this is not as bad as RPE or AMD. So okay. Still some treatment is available.
Yeah. Okay.
The other thing I would like to say is, you talked about gene therapy, but it's prohibitively expensive, isn't it, for RPE 65? I think it costs close to $1 million.
Yeah. So there, I've seen papers which debate about the cost and the insurance providers and so on. I mean, I'm not the right person to talk about the pharma, the industry-related pricing on these desperate patients.
Again, we are looking at just one gene in that case too. And
Yeah.
as you explained, that RP is such a complex disease, involving multiple of genes. It can be autosomal dominant or autosomal recessive. It could be X-linked also. It's a very complex disease.
Yeah. So eventually to take off, especially in developing countries, one needs to scale up of all pricing, everything. It has to come down. Yeah.
And you touched upon the number of photoreceptors, in hundreds of million. I think it's all ballpark figure, 120 million plus. But you also said they achieve a spatial resolution of something close to 200 nanometers and temporal resolution of 60 hertz. Can any artificial retina come close to that?
Yes. So there has been, from the hardware side, in terms of getting electrodes patterning, there has been quite a big advancement. Okay? And also patterning nano semiconductors, nanoparticles. There has been, the density has not been an issue. Okay. The spatial resolution, but in terms of the synapses, in terms of timing, getting everything right, one doesn't know. Okay.
That is very.
So I will show you some data on from my lab on some of these aspects.
Correct. See, if you look at the retina, vertically disposed cells, you can easily explain photoreceptors, bipolar cells, ganglia cells. But the horizontally disposed cells, like horizontal cells and amacrine cells, how many? More than 30 different types of amacrine cells. They all modulate the signal.
Correct. Yeah.
Would the artificial retina also take that into consideration? Providing the optimal signal?
Correct. So that is why if there is more and more single cell genomics, if everything has been sort of understood in terms of functioning, one can really use this big data ML approaches, and do a lot of processing outside and pass it on to the RGCs, That is, I like that from a scientific point of view, that's a doable thing. But again, one doesn't know what kind of.
I agree with you. In the absence of nothing, you are providing something for a blind person. That we need to appreciate.
Correct. So I see some more questions on the chat box. So there have been clinical trials for subretinal and epiretinal. And patients have benefited because the hospital and the research labs have taken a lot of effort. And there has been a mild, improvement in their vision in terms of mild, very mild, but in terms of actually people reading or actually able to see is very far away. I don't think that has been done, as far as I know,
the other question is interesting. I want to ask that too. Any device directly connected to occipital cortices, bypassing the diseased retina? You mentioned about that, but
correct. So that is another approach which where you need to again understand retina completely in terms of how you are going to give the issue out there is you need to again target, you need to enter the deeper side of the brain with all the right electrodes and do the mapping. The projection has to be done. So that is really, I'm sure it's one of the hot topics on how a difficult problem to solve, and it is an important problem to solve. And there are groups who are doing it, leading labs who are actually taken up this particular challenge. Okay.
Yeah, I do agree. LGN also could be a good area to work on.
Yeah.
See, other thing is artificial retina helpful in the cure of inherited retinal diseases? Yeah.
Yes. So there have been two of these companies who claim that the inherited diseases have been like retinal pigmentosa has been targeted. The Pixium is the AMD, but the Argus initially was going after this pigmentosa. So in principle, electrodes can help out when your photoreceptors are gone. Okay. So, so what I'll do is I will, so the two reference, so there is a reference which is available, which I just showed. It carries the details of the clinical part also. So there is a question related to that. So my talk, which is also going to be on YouTube, can be viewed again for those references. Okay.
Yeah.
So if it's okay, I can continue.
Yes, yes.
Huh?
You can have a break and drink a glass of water.
Good idea. Yeah. Okay. So
Really enjoyed the talk. It was nice.
yeah. So yeah. So we so people had asked about the materials in the lab. Okay. And so this is the sort of a bread and butter material for our lab here at JNCSR. So what you're seeing is in organic semiconductors, okay? And these, I'm calling it semiconductors because there are electrons available which are delocalized. So these are polymers which are extended on both sides, right and left. And what you have is an alternate double, single, double bond. These are the pi electrons which can sort of communicate with each other and have an extended pathway. Okay. So unlike metals which have these free electrons, you have these pi electrons, which if they are ample, okay, and they can be the you don't have lesion injury, and you can really soften the blow as they enter the soft tissue. So this is a diagram out here in terms of modulus. If you don't have these soft coatings, you go through a step function in terms of modulus. But if you have coated it with these soft materials, you can, go in a graded manner. Now, so apart from the structural aspect that these are polymers, they are also electrically active. So you can use these needles with these soft coated conductors to either stimulate or extract the signal. Okay. And another important thing is when you have rigid needles like silicon cantilevers and all inserted, you have actually a much larger impedance because you create gaps. You don't really conform to the tissue of interest or the cell of interest. So in the electrical terminology, you have much larger impedances than what one normally would see with a conducting polymer. So these are some numbers out here. Okay. So just to give you another small motivation, these are not too different from the retinal chromophore, which we saw in the natural receptor. So they are more, natural in terms of how you can integrate it. So in our lab, we have also grown stem cells on these conducting polymers. Now, these conducting polymers were on rubber substrates, and we have pulled them apart, and different rigidity gives you a different environment. And we have seen that the differentiation of stem cells into a different type of neurons can be controlled by the extent of rigidity. So there is a way to pattern cells. So the key message out here is neurons and the cells of interest not only survive in these kind of substrates, they also seem to like it, okay, and grow at a much better rate than what you would normally grow in the lab in vitro situation, okay. So, and you can control their growth. So this is unrelated to the retina, but I just wanted to say that these materials in general are biocompatible and quite friendly. Okay, so we have been able to see these neurons actually taking the cue, and in fact, the neurons very interestingly see a crack in the substrate and can use that as a trajectory to grow along the crack. Cracks which can't be seen with electron microscopy can be seen with these cells, okay, and they can use this crack patterning to grow on this. Okay? So this was a very early observations of what we had done, and we had not looked at the function. Okay, so I should just mention this as an advertisement for my field. Elon Musk has another company apart from Twitter and Tesla. The company is Neuralink. They make electrodes, okay, and one of the tricks in their electrodes is to have this P dot, these conducting polymers. Okay, so there's a paper which is single authored by Elon Musk, which carried this particular information. Okay, so the brain-machine interface again with high-density electrodes, it's a really, I mean, there are a lot of holy grail important problems out here in terms of targeting many, not only brain disorders, but also augment, you know, and do many fancier things to the brain. So in my lab, we had started this around a dozen years ago, nearly 2008-9 onwards. When I mean, earlier than that, were working with the molecular aspect, but with the whole retina, Vinny was the first student who took up this problem. I'll just show our results. Vinny right now is a faculty in the Indian Institute of Science in the Sense department. Then after she finished, Deepak joined and he just submitted his thesis on the subretinal aspects of the implant. And Abhijit is doing his PhD currently. So these three students are done the bulwark and I mean, really been doing trend-setting work in this particular field. Okay, so just to give an idea to the clinicians or non-materials people, these materials come in a bottle where you can easily dissolve it in some simple organic solvents, and you can simply coat them. Okay, these are non-patent, but you can easily pattern them using an inkjet printing technique or many other printing methods. And what we do is we take two of these bottles, okay, two different types of semiconductors, mix them up, okay, and form a film where the phase separation happens at nanometer scales. So what we are doing it in one shot in creating millions of heterostructures. So heterostructure means there are two semiconductors with two different band gaps which come close, and any interface of this kind really is very useful for charge generation. So if a photon is incident and is absorbed, an electron and hole is quickly separated at this junction, and they can have their own trajectories, okay, resulting in the electrode getting charged up very easily. Okay, so we had done this experiment to show that you can take a substrate coated with these semiconductor blends, and on the surface you will have a potential which will respond to light in a very nice manner and give you sort of a local battery, so local electrical potential available to you. Okay, and this is the media which is there in the eye. The same solvent can be used, and it survives it. Okay, and at low light intensities it works. So unlike the previous techniques which were there in the explants, which I mean the prosthetics, we don't need a battery, okay? And these absorbers are sort of the batteries, okay? They don't need to be powered separately, and they can directly stick on the region where of interest, okay? And so they can stick on the epiretina or on the subretinal part, okay? So what we do is another important thing is that we use a technique called microelectro multi-electrode array recording. So these are like out here 64 electrodes, okay, and these are connected to an amplifier. And we coat these electrodes with these polymers inks. In this particular case, it is the epiretinal, and then we bring in the transplant. And we do the recording at all the 60 electrodes. And so this is sort of a modification, not a crude form of the technique which is followed in the neuroscience lab, where they look at single cell patch clamp. Okay, so instead of looking at single cell, we are looking at the external part of the cell and doing the recording. So these are called extracellular recordings, which are good enough. Okay, they get clipped off, the signal gets clipped off, but they are available. So there are spikes, which are the most important signals, which carry the information, and they are recordable using this massively parallel approach in one shot. And there are, Dr. Raju asked, the electrodes can be made small enough. In our case, they are small enough that we can map it to a single cell or a single neuron, okay? And talk about each electrode in terms of a single neuron process, okay? So where do we get our eyes? Now we get our eyes from the chick, okay? The chick embryo, which is sort of incubated in our lab at different stages. So all animals, just like human beings, they grow their eye in a very gradual manner. So day 12, day 13, day 14 in the egg form is where the chick eggs, you have the skeletal part of the circuit available to you. And this skeletal part of the circuit is used as a blind model for us. So rather than going to either an old case, dead or close to death, this is an easier model, or go to a mouse and make it blind using either genetically modified approach or saturated with light. This is a more elegant model where we take a developing chick and introduce our explants at different stages and see whether we can make use of the circuit. Okay? So, and birds, by the way, have a much better vision than rodents for us. Okay, so mouse is the standard model, okay, for behavior and all experiments. But an avian model for retina is really easy as far as physicist background or people who don't deal with animals day in and day out. So what we do is take out the embryo, the retina from the embryo. This is what Winnie had done. She had coated the polymer on the electrodes and had light on it. And this is the eureka moment. Okay, so when we record it from the electrodes, we start seeing a typical activity like a brain activity. In this case, the signal from the amplifier, which could have easily gone to the chick's brain. Okay, and this is what we recorded. And this was, we made sure it is not an artifact by doing variety of experiments. And so the whole game now is to make sure that our signals are strong enough, much above the noise level. They are robust signals which correspond to the polymer absorber and not some remnant features of the bird retina and so on. Okay? So the retina bird is blind, and we get signals which correspond to the light. And the light, we can change the intensity, and these spiking patterns will be proportional to either intensity or the pulse duration or the background light. Okay? So there are a lot of parameters which are thrown in. Okay, the wavelength, the pulse, the background light. You need a light adaptation which needs to occur. So these were the first signals. Okay, so we were not trying to see what exactly the bird is seeing. We wanted to make sure that the bird, there is some information which is light induced, which is going into the optic nerve. Okay, so that was the first part of what happened in our lab. And then when Deepak came along, he asked the next level of question. What does the spike patterns mean? How does the retinal other circuit come into play? So instead of the epiretinal aspect, he took the polymer film and placed it where the rods and cones were. Okay, and he wanted to see whether these circuits actually mediate the signal. So again, this long painstaking experiment where things had to be repeatedly done. okay, over several years and so on. Okay. So the question, what does the chick's eye tells the chick's brain? Okay. So for that, we need to know some very basics of the computational neuroscience. How does the encoding happen? And how does the decoding happen? So there has been quite a bit of research in these areas as far as the retina was concerned, the functioning retina. Okay. So we had to learn quite a bit of it. Okay. And then we went back to our chick. Okay. So the chick out here, as I said, the embryo 14 to 18 gives us a window to see how the circuit is developing, how these cells are making the synapse, and how we artificially engineer these signals and what kind of changes which we see. Okay, so I told you about optogenetics, where we make each cells respond to light. What we are doing it, making the neighborhood respond to light and have the circuit response to it. Okay, so this is the sort of experiments we carried out. So we made sure that everything was, so all the normal protocol which needs to be done in a physiology lab, we have done it. Okay, and in the interest of time, I just want to show you the kind of complexity which is involved. Okay, so there are, I mean, huge number of signals. Okay, recordings, and these have to be analyzed. So Abhijit and Deepak went about doing it quite systematically. The first involved the spike sorting. We had to pick up the spikes in the sea of signals from each of the electrode, quite a bit of data processing with a lot of conditions, preconditions, so on. But we made sure we didn't lose out any information in the process. And the next thing which we wanted to do was classify the cells. Okay, so the classification, which is normally done based on structure or genomics, we wanted to see whether our functioning can actually classify the cell. So we have the different on and off pathway. Okay, so I just want to show one example, okay, of classification of these cells. What we are doing now is we have a pulse of, a light pulse, and we are recording. Okay, and what in these recording is we are picking up the spikes exactly repeatedly from the region. So what we are seeing is when the light is on, we get a spike from many of the electrodes at the same time. Okay, repeatedly and repeatedly, and then we do this spike histogram, and then we count. So after these repeated trials where we know the precision was quite important, we actually plotted in a manner where you can see that there's a clear histogram in the region where the light on cells. Now there is another class of cells which fire where the light is off. Okay, and you can see that these are the off cells. And then there was another class of cells where both on and off. So based on a simple light stimulus, just having pulses of light coming in and light on for a short duration and light off for a reasonable duration, we are able to classify. Now there is a lot of parameters out here, the external parameters which involve the pulse of light, the distance between each of the pulses in terms of the timing, and the background light. So each of these parameters can be taken as a descriptor in our problem and do this cluster analysis. Okay, so this cluster analysis is really useful in telling us also, as the days evolve, how does the circuit evolve. Okay, and if the eventual question of looking at the mature chick and comparing it with a blind chick which has got this transplant in it. Okay, so we have to do these experiments on an adult chick after the chick has come out and hatched, and we look at that also quite in detailed manner and do the signal analysis. So there is another important protocol in this whole analysis is in order to identify the different pathways, we can use the biochemical approach where we can introduce chemicals which block the pathways of choice. So this again, we made use of the extensive literature which was available, and we can really target these on cells and off cells because the mechanism is known on how they come about, and we can block those particular, in this case, the glutamates which are involved, and with the blocking agents, which are ionotropic and metabotropic. Okay, so these are, I mean, again, very standard protocols which are there in the field, which can be utilized to give you, I mean, these are necessary for getting a paper published, okay, apart from the fact that this is an important thing, okay. So it is quite a big of lot of hard work out here in terms of both learning and actually implementing it, okay. So both Deepak and Abhijit really went through the grind and did these difficult experiments. These still are going on, and we were able to get these kind of circuits and pathways based on these blocking agents. Okay. So again, I have to walk you through the data and convince you, but I will give you the reference for our recently published paper and the one which is there in the archive for you to go through this. Okay. So, I mean, you have to believe me for the time being that these artificial the polymers really work in terms of generating signals which these blind chicks are able to process at the retina and give out these information, these spike patterns. And another important thing which we saw, which is the next level of intelligence, is the spatial correlation. Dr. Raju mentioned in terms of spatial density. So we can actually look at the neighboring electrodes and see how they talk with respect to each other once they have received the signal. Okay? So we have seen these, we do this cross-correlation analysis between these different electrodes, and we can again tell that where these, depending on the extent of the correlation, we can tell you which circuits are in place in terms of the horizontal circuitry, which was mentioned earlier. Okay. Okay, I'll just show you that. Okay. Okay, let me see which. I don't have this horizontal. I'm sorry. I think I missed out an important slide. But we can, based on the spatial correlations, we can tell you that the horizontal circuits are in action at the outer plexiform layer and the inner plexiform layer. The different depths of the signal processing could be envisaged from the signal analysis. Okay. I know it's getting late in the day for me to give you real super technical detail, but you have to take my word for granted. Okay. So what I've just told you right now is in our journey of getting signals out artificially and making sure it is something real, what we have done is looked at a mature chick, okay, which is where the eye retina is intact, and done the signal grabbing and the analysis in detail. I mean, reasonably in depth. And then we go back and check it with the day 12, day 13, day 14, 15, 16, 17, 18. Okay, 19 is when it hatches. Okay, 19, 20 onwards. So compare these different days and look at these signals and compare with the mature. And we see a lot of similarity. Okay. I mean, as far as the stimulus, flash stimulus is concerned, there are other patterns of light, different colors of light, video, moving images, three-dimensional, and there are other features, okay, of light where other circuitry are involved. So those are natural questions to ask and for us to figure out. But hopefully we have a sort of a roadmap in front of us on what is the near future, okay, and what are the short-term goals and the long-term goals. Eventually, we need to enter into a more higher form of animals and enter into a clinical lab where we can work with patients. Okay? So that's a long road ahead, but hopefully this talk elicits attention and some questions and people coming back to us, and where we can collectively use our complementary expertise, okay, and form a community which can take us to the final goal. Okay, so thank you again, and if you have questions, please do come back to me. I will try to take it for you. I can give you my number in the email, and we can talk. Again, thank you very much for staying back. Looking forward to the questions. Thank you, Dr. Raju.
Thanks a lot.
Over to you.
Are we still live?
Yeah.
Yeah, I have some 4 questions. Would you like to address them?
Yes. Yeah.
First question, how do we choose the material for artificial retina and its biocompatibility?
Yeah. So
It was for the selection.
Yeah. So what we do is we have a secondary cell line available to us through my colleagues out here. We have a cell culture facility. We grow these secondary cell line on these different coatings, and we see their, growth, proliferation, and all the parameters. And if things are okay, we move on to the next level. And then the other thing is we also can look at, there could be a more detailed way of looking at it from the toxicity point of view. There are, again, very well-established protocols. Once you put the primary explant, whether the substrate is doing any damage to it, both mechanically or electrically and so on, over a few hours, we can monitor it and see whether, again, with a lot of controls, and seeing whether it is functioning in the way it is functioning. Again, comparing it with the mature chick. Okay, so there are a lot of indirect methods, but all put together, we know that we are dealing with material semiconductors which are not toxic. So I should tell you, there are a lot of other semiconductors, okay, which have got inorganic materials, lead in it, all kinds of cadmium and so on, which are neurotoxins, and that has been very well established. Okay, so I wouldn't want to go near those, and since I want to promote the material which we know reasonably well and understand, we want to stick to these materials which can be grown both in a film form as well as in nanoparticles, and it can be injected into the region as a sheet, So.
Yeah, it's interesting. The other question is, why you use a polymer, which is a semiconductor?
Yeah, so if it is not semiconductive, it will not be absorbing the light. An absorption of light deals with the fact that there is a band gap or a transition which involves the pi electron, okay? So, which is available for setting up the surface potential. So you need these, the light-induced voltages and currents. That is why we used polymers, which have strong absorption, and these absorption strength are very comparable to the retinal, the chromophore, natural photoreceptors in terms of their strength. So we use, again, very nanometers, 50, 60, less than 100 nanometers, where it is still transparent and you can still have strong absorption. Okay, so there are a lot of science fiction gadgets can be made. We have done it in the lab with these, that your glasses can see things and you can transmit it and you can get back information and so on. But this can be injected into the retina. Okay.
Yeah, it's truly a fascinating work. I have a question. Do you come across any action potentials also?
Yes. So So
There seem to be very high frequencies suggesting that more of graded potential.
yeah, so I have not done this. There is this Italian, not friends who are sort of also have their own project. They use the single semiconductor while we use a blend. They have actually looked at the single cell patch clamp techniques based on these coatings. They've also done the in vivo measurements and shown that it works to reasonably well. Yeah.
And another suggestion, when the eyes are donated to us, we use only the cornea. Retina is available, human retina.
Yeah.
They're viable for a few hours.
Yeah. So this is because of the recording methods. We have if you do the perfusion over a very long hours, we can keep it alive. Okay. And with all the reagents and all the fuel and the right fluid composition, you can keep it alive for a longer period. So
yeah, with the DME
shown. Yeah. So we have from our lab, we have been able to preserve cells over several days. Actually, Anil Krishna, who unfortunately is not with us, who passed away. He had come up with a cell assembly where we are able to prevent an oxygen bubbling around these cells and can make it survive for much longer duration. This is again a published. So we haven't put it together, all these different components within my own lab.
And you are going for a short-term recording anyway.
Yes, we are going for
get the data in a short term.
Yes. So there we have enough time. So the only thing is we need, good hands and good days to happen, and the chick egg should also be healthy enough. There are statistics. These are not material science experiment. These are biology where life is there, and statistics is also a very important factor. So so doing it repeatedly is quite one has to enjoy it. It's difficult to do that, but keeping in mind the motivation that you're working for an important problem, that is what keeps these students going out here.
So you asked the question, are we there? We will be there.
How?
You asked the question, are we there? My answer is
yes. So no, we are not there, but we have good promise, and with the support of the community and all the funding agencies and some collective synergistic action, we can see it within maybe, I don't want to give a number, but if everything goes good, if not us, things are going to happen within a decade. The coming decade, things are going to happen. It is not audacious as people claim it to be. It is things are in place, and people understand.
I said we will.
Yes. So
science progresses in small increments anyway.
Yes. So, and there is a question related to modulus. Okay, how do you measure modulus? I don't measure modulus. But this is data available in the literature. A couple of my colleagues in US who do it. I have worked with them, and there is a rheometers available, and people do these calculations and finite element methods. And look at the way the indentation and all makes a difference to the surrounding, and people have estimated this quite well. Anything else I'm missing out? Or
from my side, I think everything is fine. These are the questions. And you have really mastered the photoreceptors and bipolar cells. Now we will move to ganglia cells. Whatever input we can give from a biologist or a physiologist point of view, we'll be happy.
Oh, thank you.
Thank you very much, Narayan and Raju. I think this was an absolutely fascinating discussion. We've really not done much on biology in this webinar series. So, we've been looking forward, Narayan, to this webinar for a long time now. So thank you so much for doing it. And thank you, Raju, for moderating today's discussion.
Welcome.
So just a few takeaways and a bit of summary of what happened today. We started by looking at the eye, an absolute marvel of engineering. When you look at everything that's gone into it, something that consumes so little energy and yet does so much, can respond to a single photon. Our cameras are nowhere near any of that as yet. Every part, Narayan mentioned, every part, every little bit has a meaningful and very important function. And the architecture is extremely robust, and this is something that we've not been able to replicate in anything that we've made so far. We then understood the concept of what is involved in seeing the whole cascade of the enzymatic reactions that go into the process of our being able to translate something that is in front of our eyes into our brain and therefore see. And then we looked at basically today's discussion focused on two retinal disorders, retinitis pigmentosa and age-related macular degeneration. We looked at various ways of therapy. We looked at gene therapy, which is possible at the early stages, but is awfully expensive. Probably Elon Musk could take us to outer space and back in less money than what it would cost to have this therapy done. Optogenetic and stem cell therapies at later stages. But again, there has not been a very high rate of success as reported as yet. And therefore, the importance of retinal implants. Research on all of this has been going on for perhaps about 3 decades, and placing different kinds of implants, different technologies near the retina, near the brain, and so on. So different models have been tried out. The best that we've so far got to seems to be, vision of 20 by 500, which is with the Prima mechanism, which is an external camera with photodiodes implanted on the retina itself. Arbus 2, which is an implant on the retina, still gives an effectiveness of only about 0.9%. So very, very, very far from where we ought to be for any of these to be viable. And then we looked at the work that's been happening in Professor Narayan's lab over the past 12 years, is thereabouts? Or?
Yeah.
Yeah, and he talked about, he gave us, I think, a very, very detailed description. And thank you so much for sharing all this, Narayan, so openly with us. So, so nicely with us in so much of detail. Just a few things. One is he talked about how they mix different kinds of semiconductors and polymers, and how that arrives at different semiconductors that can be used to coat the substratum. He talked about the whole idea of bulk heterojunctions, the fact that it's tiny enough, the micro electrode array is so small, and that it can be mapped to a single cell or a single neuron. So it's and therefore it can stick on the retina or in the either retinal or subretinal areas. And of course, it needs no external power, so it's something that works completely on its own. And then he talked to us about how they've been able to develop this using the chick's eye as it is developing. So how exactly is the retina developing, and therefore how do these things work? And then we looked at encoding, decoding, and how they happen in the final, in a functional retina, and therefore translating it back. he talked about spike sorting and then classification of cells and on all of that data analysis and what it implies. Finally, it talked also about the biochemical approach to blocking certain pathways so that you can target specific on and off cell. But the basic takeaway from today's talk has been that polymers do work. And we have actually seen the data that tells us exactly how the spikes are created and how they are transmitted to the brain. And then the final comparison between the normal mature retina and a blind retina together with a polymer prosthesis, and what exactly is the comparison between these. We saw all of that. And I must say that we are definitely looking forward to all of this research translating into something for people who are suffering from these various conditions. Probably other conditions as well will get addressed once we see this technology actually moving into practice. So we look forward to all of that, Narayan, and all the best to you and your lab and the work that you're all doing. Thank you ever so much. And thank you to our audience for being with us today and for your questions as well. We look forward to staying in touch with all of you. And this recording will be up on our YouTube channel come Monday, Tuesday. Please do feel free to share it with anyone who might find it interesting. And we look forward to seeing you on our next webinar. Okay, thank you very much.
Thank you, Sushi. Thank you, Dr. Raji. Thank you, Abba. Bye.
Thank you. Bye. Bye, everyone.
It was a great evening. Thank you.
Thank you.
Thank you.